Hello, everyone. Thank you for coming today. My name is Erika De Benedetti, and I will manage this webinar. Before to get started, I would like to give you a couple of information. Firstly, this webinar is recorded, and the recording will be available on biostimulant.com website and on our social media in the next days. Secondly, I invite you to use the Q&A box that you can find at the bottom of your screen to submit questions. We will collect your questions and answer to at least some of them at the end of the presentation. The topic of today is application of seaweed extract as plant biostimulants. It's my pleasure to introduce our speaker, Arup Gosh. Dr. Arup Gosh is Senior Principal Scientist and Divisional Chair at Applied Phycology and Biotechnology Division of Central Salt and Marine Chemicals Research Institute at Gujarat, India, and Professor of the Academy of Scientific and Innovative Research of Ghaziabad. So, welcome, Professor. The floor is yours.
Thank you, Dr. Erika. Also, thanks for the nice introduction which you gave. Am I audible?
Yes, we can hear you well, see you, so you can start.
Okay. So today I'm going to give you a brief talk on application of seaweed extracts as plant biostimulants, and I'm very happy and I'm very thankful to you, Dr. Erika, for giving me this opportunity to speak in this forum. So I may not actually introduce seaweeds. Most of the people will know seaweeds. But to introduce, I would like to first introduce our institute, which is coming under the aegis of Council of Scientific and Industrial Research. So this is the largest publicly funded government research organization in India. And we have about 37 different laboratories spread across the whole of the India. And we are located in a place called CSMCRI Bhavnagar, that is in Gujarat state of India. And we are one of the 9 top CSR institutions, and also we have a ranking in the top 650 institutions in the world as per the SciMago institution ranking. So we are called Central Salt and Marine Chemicals Research Institute, CSMCRI in short. What we do in our institute, other than seaweeds and seaweed biostimulants, we do anything that is related to seawater. So that includes research on salt, research on desalination of water, different types of desalination technologies and water purification technologies like nanofiltration, ultrafiltration, reverse osmosis membranes research. We also do a lot of research on marine chemicals, mainly deriving chemicals from the sea, natural products from the sea, and marine elements. Apart from that, in biology, we also do not only seaweeds but also microalgae, halophytes, salt-tolerant plants, including the biotechnology. We also do research on marine microbes, and the aim is to derive marine bioactives from them. Other than this, we have a very strong presence in the marine pollution sector, and where we do environmental impact assessment. And recently, we have also started some in-depth research on microplastics. So with this introduction of what we are, our motto is to explore, harness, and transform the marine resources for the good of the people. We at CSMCRI not only do seaweed biostimulants, but also we have the upstream technologies for cultivation of different commercially important seaweed species. For example, you can see Geladiella acerosa, Enteromorpha flexuosa, Gracilaria dura, Gracilaria edulis, Gracilaria debilis. This figure, you can see that We have developed technologies to cultivate the seaweeds in the sea. This is particularly the photograph of seaweed that is called Capophycus alvarezi. This is a red seaweed, and other than this, we also have this Debilis and Edulis cultivation technologies also developed. In seaweeds, what we do is we develop cultivation and seed production methodologies, develop commercially viable value added products including biostimulants, animal feed additives, and the phycocolloids like carrageenan, alginates, agar, agarose, and so on. By doing this, we also help capacity building of the village fisher folks who are mainly engaged in seaweed farming, wherein we have also developed techniques for macro and micro propagation of the seaweeds. uh, my topic is more about the plant biostimulants, but just to touch upon, I would like to inform the audience that very recently, in the last 2 3 years, we have developed animal feed additives as well, and we have in fact licensed these technologies as well. What are the salient features that I can say is that we have developed animal feed additives based on different types of seaweeds, which have shown improvement in the performance of poultry as well as cattle. Poultry comprise both egg layers as well as the broilers. Better immunoresponsive cellular mediated as well as HA titer immune modulation in poultry and cattle. We have found that gut health, both microbial and structural gut health in poultry is improved. Higher egg production and advancement in egg laying age. In case of milch cows or the dairy animals, we have found that the although the milk quantity is not increased, but we get a higher calcium and iron content in the milk. Most important with seaweeds is the reduced methane emission and higher energy use efficiency in animals which are fed with such sort of seaweed based animal feed additives. We have also found higher daily growth rate in the crossbred calves. Many of these formulations we have also undertaken toxicological studies, and these were all done in collaboration with the best veterinary, avian, and dairy research institutes in the country. That is again under the aegis of another organization that is called Indian Council of Agricultural Research, along with the toxicity trials that we in our sister institute located in Lucknow, that is called Indian Institute of Toxicological Research. Uh, now, coming back to the topic, that is use of seaweed biostimulants in agriculture. Seaweed biostimulants has been used from very long time in the country. But earlier, about 2 decades back, 100% of it was imported. But with our intervention, we tried to develop biostimulants based on the seaweeds that can grow in Indian tropical waters. So whereas earlier we used to import the maximum of it from the countries that are having cold sea. For example, Ascophyllum nodosum dominates the market. Litho, Thamnium, Calcerum, Macrocystis, Eclonia, and Dervilia. These are the species which usually are imported. But we have developed technologies for deriving the seaweeds from the tropical waters as well. And we have actually done a lot of science behind the working of biostimulants. We try to unravel how these seaweed biostimulants work in soil, in plants, how it can help abate the climate change impact. Then just a piece of information that earlier in India, biostimulants were not regulated, but now the government has recently initiated this step, and now it is regulated under the fertilizer control order. And right now, they have specified, government of India have specified certain specifications certain minimum, certain maximums which should be there in the biostimulant, which should not be there in the biostimulant. So we are working on these procedures as well now. So coming back, we have developed the technologies. We have IPR secured for these. This is the sort of biostimulant from, I mean, Carpophycus based biostimulant. And these are the pictures how the entire value chain we can get not only the biostimulant but also the carrageenan from the same seaweed biomass, which is actually used. In this carrageenan is an hydrocolloid, and this is used in many products across different food as well as pharmaceutical sectors. For example, in toothpaste, in ice creams, in pet foods, in clarification of beer, and so on. Presently, the Indian market size of seaweed biostimulant is about 1500 kiloliters of liquid formulation and 4.5 lakh tons of solid granular formulation. But I would stress here that to our best estimate, even now 10% of it is indigenously produced, 90% of it still is imported from other countries. And the bottleneck is like seaweed cultivation, seaweed biomass. Now we have validated the results of the biostimulants on different crops. So towards this, a multi-institutional, multi-crop project was coordinated by our institute, the CSMCRI, wherein the efficacy of two of the seaweed biostimulants derived from one Carpophycus alvarezi and Gracillary edulis, they were tested and validated at 43 different state agricultural universities across the India, and we did it in 20 states of the India. 20 states of India, and you can see these were multi crop trials wherein we collaborated with different agricultural universities located in different agroecological and agroclimatic zones, different types of soil, and then we gathered, we collected a lot of results, and we analyzed the results, and finally, for example, what you can see is that in rice crop, which is the most important crop of India, we did it on more than 20 places, and we found modest improvement in yields of the crops in rice, in maize, green gram, black gram. These are pulse crops, sugarcane, and soybean, that is the oilseed crop. And this database generated actually aroused a lot of confidence among the stakeholders to market the biostimulants under their different brands. We have already licensed these products to several of our licensees. But what actually matters is the science behind it, and which is what we did at CSMCRI, the science of the biostimulants. So, if I were to tell you the results of the biostimulants, this pie chart, which you can see in the left part of the screen, we did, for example, rice 38 places, 38 places we did rice trials, agronomic trials, and we found an average improvement of about 18.7% over and above the recommended dose of chemical fertilizers. Similarly, for maize, we did trials at 23 different places, and we found that more than 20% improvement in crop yields. For pulses and oilseed, especially, the yield improvement was always more than 25%. For example, green gram 26%, black gram 36%, sesame 26%, and soybean 36%. So that was the, I mean, and these were averaged across several areas, several places. We also found a good improvement in potato and sugarcane, where is about 16 to 17% increase in sugarcane and potato yield was found over and above the recommended dose of fertilizers. Now, what was the salient research outcome? So we found ranging from 11 to 37% increase in crop productivity, In several of these experiments, we tried reducing the dosage of the fertilizers, and we found that 25% reduction in the use of recommended dose of chemical fertilizers is possible. Then we also did a lot of experiments wherein risk mitigation against climate change was assessed, and we found that the use of the biostimulants, particularly I'm talking about the Capophycus alvarezi biostimulant, it imparted biotic and abiotic stress tolerance. I would come to the detail in the future slides. We also quantified beneficial environmental impact by life cycle assessment. Then we also went on to prove the benefits of the use of of the biostimulants to the soil bacterial community. But I mean, apart from all these, why it is important? I mean, the seaweed cultivation itself, the cultivation process itself, it gives us, I mean, enhanced income and rural employment opportunities, especially to the women. Then we find local employment in the form of the industries which are set up based on this. Entrepreneurship is something that is very important. Then I mean, the products, when it comes in the market, it also helps in local skill development. And the most important thing is the farmers who use the products, they get enhanced crop production, their income is increased at a low carbon footprint. Our estimate is that in the last 1.5 decades, only by our interventions, more than 2,000 million euros. I mean, the farmers' enhanced income has been increased by 2,000 million euros. And we have been also recognized by different government agencies for the initiatives which we have taken. So I was talking about the Capophycus alvarezi. Similarly to the Capophycus alvarezi, there's another seaweed called the Gracilaria edulis, and similar to that, we have undertaken extensive trials in, again, rice, green gram, black gram, sesame, soybean, fodder crops, sugarcane, potato, and we found a significant improvement like that. I mean, in that case also. So not only Capophycus but Gracilaria edulis also improves the crop production or the crop yields of different crops tested across multiple locations in the country. So in all, more than 175 trials each for each of these biostimulants, along with another 175 trials of demonstration level trials in a larger acreage was also done, and we found very encouraging result. The third type of biostimulant is, I mean, the earlier two, Capophycus and Gracilaria, belong to red seaweed category. We have another seaweed that is called sargassum. There are many species of the sargassum, like Sargassum whitey, Sargassum cinctum, and we have the technology to develop biostimulants from them as well. And we tested it also in different crops, and we found it good in the spice crops, onion, as well as groundnut. So we found a significant improvement in yield using this brown seaweed biostimulant as well. One very important findings we found is that when it was applied to the apples, the shine of the luster of the apples was improved, and this has been done in the state of Jammu and Kashmir, where apples are grown in India, and they are very good quality apples. Now, coming back, what we basically do in our institute is the science. So we did a lot of experiments in the institute in order to, I mean, have a fundamental understanding on mechanistic action of the seaweed based biostimulants. And for different types of experiments, different objectives, we did the detailed study, and each of them have been converted into high impact papers that you can see that has been published, and it is on the screen. These are all, I mean, publications from our institute by the group of researchers who are undertaking these studies. So overall, you can see that we have, I mean, cleaner increase in crop production over and above the recommended dose of fertilizers. In cereals, we saw that number of tillers or sets or starch and sugar content especially improved. In pulse crops, we found that the number of pods and number of root nodules improves. invariably there is an increase in root volume and root length by application of the seaweed biostimulants. We can reduce the inorganic dose or inorganic fertilizer dose by 25%. We also identified the best responsive crop stages for achieving maximum efficiency. Now, this is important. Why this is important is that it is actually the stage at which you should one should apply the seaweed biostimulants is stage specific. This is what we found in maize, wherein we tried to give different combination of treatment at different leaf stages of the maize, and we found that I mean, if we are to combine, so we can identify the best stage, the best two stage, best three stage, and which are the stages at which the biostimulant does not work. For example, when it is 5 leaf stage, 10 leaf stage, and 15 leaf stage, if I give the biostimulant spray at 10 leaf stage, you really won't take you we don't really get significant improvement in the yield or the quality of the crops. But if you give it at 5 leaf stage and 15 leaf stage, you get the good results. Even it gives good results if you have only one spray, one opportunity to give the spray to the crops. You better do it at 5 leaf stage, and the effect is I mean very good. But if you have higher number of applications, then I mean depending on the most effective stages, crop stages, one can identify and give. So we have done only for maize crop for this, but I mean, uh, uh, but there are critical stages for application of the seaweed biostimulants in all the crops that we are taking care. Then what is the what are the bioactive compounds and what are the mode of action? So this also we have studied in detail, and we found that the seaweed biostimulants contains a lot of plant growth hormones, natural plant growth hormones like auxin, gibberellin, cytokinins, quaternary ammonium compounds like glycine, betaine, and choline. Glycine, betaine, and choline are actually the one, I mean, the chemicals which also impart plant stress tolerance. So we did a I mean study on the roles of these quaternary ammonium compounds, that is glycine, betaine, and choline, and we were able to find their importance in abiotic stress tolerance. We also did a quantification of carbon footprint. Like in order to call any biostimulant green, you actually need to do a elaborate life cycle assessment, that LCA approach you have to follow, which we did, and we found that per kiloliter of the seaweed biostimulant production, the carbon footprint is only 11.6 kg carbon dioxide equivalent. So that is very less compared to the inorganic chemical fertilizers, and that is why we say that when we apply these biostimulants to agriculture, we actually emit out less carbon per unit of food production. So this is not only important from research point of view or academic point of view, but it is also very important from policy point of view. So likewise, I would also urge that all the biostimulants should be actually, I mean, looked into from life cycle assessment approach, and one should actually find the carbon footprint. This is also important because different methods and different extraction methodologies will have different carbon footprint, and one must choose the best ones. Then we also found the beneficial effect on soil properties. We found improved soil enzymatic activity, improved soil microbial population. Several genera were also favorably influenced, and there were some abundance of some genera which are very important for soil nutrient cycling process. Even it was found to be good biostimulant for microalgal cultivation like chlorella vulgaris. Okay. Then we also did a transcriptome study. This transcriptome approach was to find which are the genes that are differentially regulated when these biostimulants are applied on the crop. So we did both root transcriptome as well as the leaf transcriptome. And we found there were several genes in different pathways that were positively modulated, and we found that nitrogen assimilation genes related to nitrogen assimilation were overexpressed. Genes related to nitrate transport were overexpressed. There were certain enzymes that were associated with DNA damage. They were also, I mean, upregulated. There were enzymes like cysteine synthase, that is the key enzyme for the production of antioxidant glutathione, both in its oxidized and reduced form, that is GSH and GSSG. They were upregulated and offered stress tolerance. Then there were key enzymes that were responsible for fatty acid synthesis that were found regulated. Several cell wall synthesis related genes were upregulated. Especially if I were to comment upon hormonal signaling, we found that gibberellic acid hormonal signaling was down regulated. As a result, the DELA proteins, these are the transcription factors, they got increased, and that led to the enhanced tolerance in case of abiotic stress tolerance. For roots, it was like that. For leaves also, we found similar, like nitrate transportation, nitrogen cycle was common, and there were upregulation in signal transmission, photosynthesis, transmembrane transport of ions, glycogen, and starch biosynthesis. In fact, under stress conditions, the processes that led to degradation of starch under stress, they were actually down regulated. So that helped in improving and preserving the starch content in the crops. As a result, higher yield was obtained. Then there were genes that were responsible for catabolism of polysaccharide molecules. Just I said, they were found down regulated. So on and so forth. I will not go very much into detail, but we also understood, we unraveled what are the antioxidant activity and which are the reactive oxygen species that were affected, and how lipid peroxidation was reduced and tissue damage was reduced. We also found that electron transport processes were improved. As a result, water use and photosynthetic efficiency was also improved. Not only for the abiotic stress, we also found studies, we found that these biostimulants can also modulate jasmonic acid and hormonal pathway, and regulated phytohormones and antioxidative activity. We found this both in Carpophicus as well as Sargassum. So there were certain enzymes like catalase, which was improved. Hydroxide radicals were also improved, I mean higher, but then proline, which actually quenches these hydroxide radicals, that was also found to be increased. So proline increased, then lower H2O2 production was found. So that's why the reduced SOD, superoxide dismutase, was actually there. Now, physiological studies also were done using this seaweed biostimulants, and you can see two lines. One is red line and one is blue line. The blue line is the control, and the red line is Carpophicus seaweed extract. So when these were applied to the maize plants, what we found, the cycle that we found, the increased photosynthesis by use of this Carpophicus seaweed biostimulant. Apart from photosynthesis, we also found improved electron transport rate, improved water use efficiency, improved FEFM ratio in the gas exchange measurements. This is photosynthetic measurements done by infrared gas analysis. Uh, so we also tried to understand what happens under drought conditions and what happens under normally irrigated conditions. So we simulated, and we found that the effect of the biostimulant is different in case of drought, and it is different in case of irrigated conditions. So depending on the condition, uh, there are modulation inside the plant. So it is not like one process or one particular type of processes happen or they follow this trend. So our hypothesis overall is that The photosynthesis process is efficient. That's why lesser oxide radicals are generated. It leads to lower superoxide dismutase activity, lower oxide radical generation, and the elevated level of catalase, the quenching enzymes, APX combinedly lowers SO2 production. And we found the increased mdhar, GR enzymes and the non-enzymatic antioxidants like ascorbic acid and glutathione, they are also improved. So what I was telling, that under drought condition, the plant behaves differently under by application of kappa ficus biostimulant, and under well water condition, this is WW is well watered to the right. And the type of biochemistry that is happening inside the plant, biochemical reaction, they are different. For example, salicylic acid, it increases by application of biostimulant both under drought as well as well water condition. But when it comes to indole acetic acid and abscisic acid, under cell drought condition, kappa ficus actually it decreases. And V5 and V15 are the stages, and it also depends on the stages. So there are a lot of variables with respect to stages, variable with respect to conditions at which the biostimulants are applied. Even I will say that the seaweeds are natural plants or natural organisms, and the stage at which you harvest and the season at which you harvest, they also have bearing on the composition of the seaweeds. And that's why I think that you ought to expect variability in this case also. So that is why standardization of the constitution of the seaweed biostimulants is very very necessary, and this is something which we are right now currently doing. We are trying to analyze the biostimulant constituents by metabolomics, using state of art equipments like LCMS, HRMS, mass spectrophotometry, MSMS analysis, FTIR studies, and so on. Now what the next we did is that we found that invariably there is a change in the root length and dry root weight by application of these biostimulants, whether it is under drought condition or it is under irrigated condition. In drought the response is high even though the yields get lower, but by application of these biostimulants, the diminishing thing can be controlled, so the decrease can be decreased. Whatever decrease has to happen will happen, but that percentage of decrease can be decreased by the use of this biostimulant under drought condition. And that's what this particular data shows, wherein we found increase in seed weight over control about 36% in case of drought condition, whereas under well watered irrigated condition the use of biostimulants could only increase by 27%. But if you see the overall grain yield, it is always higher in irrigated conditions. So what I was stressing is the percentage of decrease can be controlled by application of this biostimulant under abiotic stress condition. Now under abiotic stress condition, we also did full macro and micronutrient analysis in all the plant parts of the maize. For example, seed, stem, leaf, root I am not showing, but that has also been done. In all these cases, which you can see here, the notations are like D plus DSAP means drought plus SAP. SAP is nothing but biostimulant. D is only drought. I is irrigation plus SAP, that is irrigation plus biostimulant, and I is simple irrigation. So these are four different conditions created, use and non-use of the biostimulant under drought and irrigated conditions. So what we found is that nitrogen content in the plant parts, whether it is seed, whether it is stem or leaf, it improved invariably by application of SAP both under drought as well as irrigated conditions. So likewise iron content was also increased. So there we thought that there might be some correlation why it is happening, and that's why we zeroed in on this transcriptomics approach. So by transcriptome analysis we found You can see in this picture the green dot nitrate transporter. So nitrate transporter is a protein that is there in the cell membrane of the root cell wall, root cell membrane. So the overexpression of this or upregulation of this helps to take the nitrogen from the soil into the plant roots in a more efficient manner. Similarly, after this nitrate gets inside the plant roots, the process is like first conversion is from nitrate NO3 minus to nitrite, that is NO2 minus, and this is mediated by an enzyme that is protein called nitrate reductase. And you can see the two green dots. That means there is upregulation in the activity of nitrate reductase in the root cells, and this is the first rate limiting reaction, or the first rate limiting reaction happening, wherein nitrate first gets converted into nitrite, then nitrite gets converted into ammonia, ammonia gets converted to amino acids, and then it gets into protein. So when the first rate limiting reaction is enhanced, so that actually leads to higher nitrogen uptake. So I would again stress, like these biostimulants, if you were to really analyze the biostimulants in terms of nitrogen content, I would say it is having negligible content of nitrogen content in it. But in spite of not having any nitrogen, how it is improving the nitrogen content inside the plants? So these are the physiological and molecular process, molecular biology happening inside the plants. So similarly, we analyzed several other biochemical pathways, and and we published also. This has been published in Journal of Applied Phycology in 2020, wherein several different physiological processes like cell wall, lipids, secondary metabolism, amino acids, starch degradation, sucrose pathways, nitrogen cycle, they were positively influenced. There were some upregulation genes that were upregulated, and there were also some genes that were downregulated, but the downregulated ones were also, downregulation of such genes were also beneficial to the plants. So if anyone actually wants to really look into the details, they can always look into these papers, or anybody wants, I can always send a copy to any of you. Similarly, like sulfur metabolism, we found the key enzyme, that is cysteine synthase. Cysteine synthase is a key enzyme for the production of antioxidant glutathione, and is expected to play a role in quenching of the reactive oxygen species. So cysteine synthase was found, upregulated. This gibberellic acid signaling system is important for it is important for imparting drought stress tolerance, and this is what we actually found. There were certain genes that were that result in reducing the gibberellic acid signaling, that led to increased DELA transcript, and that enhanced the abiotic stress tolerance. So we also did a differential expression of the transcription factors, and there were several transcription factors like MAB, then trihelix, even this BHLH, and auxin related things that were found overexpressed, upregulated. This DELA actually comes under the GRAS protein. So that was that we found that it is very important in imparting drought tolerance by gibberellic acid signaling. Not only in wheat, we also did gene expression studies in tomato and wheat, and we found that it is also giving stress tolerance in case of drought, as well as this salinity. So these are initial results where we found that there are certain signaling molecules like MAP kinase genes. There were Virki genes that were overexpressed. The catalase content, catalase activity was also high, and SOD activity was also high, and these were also published by the group who did research on this. A very important thing is towards the right, you can see the work on tomato. So till now, I have been telling about the abiotic stress tolerance, which might be drought tolerance, which might be salt tolerance, and so on. But the use of biostimulants that it can actually improve resistance against the fungus, a fungus called Macropomena fasciolina, is also something that we found it does by the use of the biostimulant, this Kappa ficus biostimulant. So I will explain the color codes. So blue color is KSAP, wherein the plants which are, these are tomato plants which are not infected with the Macropomena fasciolina, which is a fungus that causes collar rot, and this is a very dangerous disease. So we artificially inoculated the tomato plants with this Macropomena fasciolina, and the red one is so the plants which are treated with Macropomena fasciolina fungus infection done, and the green is where we infected it with Macropomena fasciolina, but also gave the KSAP. KSAP is your Kappa ficus biostimulant. So what you can see the results that there are pathogenesis related genes, PR genes. These are the plant defense genes that gets overexpressed in presence of Kappa ficus sap, that gives tolerance. So for example, you can see PR3. If we only apply Kappa ficus sap where there is no disease, the expression remains low. If only Macropomena fasciolina is infected, then also it remains low. But this PR genes, in presence of Macropomena fasciolina as well as this Kappa ficus sap, it gets overexpressed. Similarly, is MPK2. So this work was done by a group of researchers in our institute, and that paved the way that seaweed biostimulants, not only for Kappa ficus, now there has been studies have been initiated for sargassum also, and we are finding similar results. Uh, I think still I have got another 10-12 minutes left. So I would also like to touch upon ecological part of the use of biostimulants. One can always question what happens to the soil microbes when these biostimulants are applied on the plants. They may be applied on foliar, they may be applied in the soil also. But we never expected that when we do a foliar spray, we'll get any changes in the soil microbial community. But just to ascertain that, what is the effect? Uh, we actually then found that about 20 to 25% of the spray that we do on the plant foliage or plant leaves as a foliar spray, it actually drifts into the ground or it falls as a drift spray on the soil. And then we did a 16S rRNA metagenomic study, and to our surprise, we found that there were improvement in soil microbial diversity and population by the use of biostimulant under irrigated condition when they were spread at three stages of leaf. B5 is 5 leaf, 10 leaf, and 15 leaf. So the green, the bottle green, the sea green is the one wherein the plants were subjected to stress. We withheld irrigation, and we also applied the this biostimulant, and then we checked the microbial population in the soil. So the control were the normal irrigation, and control were where the plants were applied with the stress by not irrigating it, but at the same time only water was sprayed and no seaweed biostimulant was done. In some of the cases, for example, for bacillus, you can see there is a statistically at par data with respect to even normal condition. Wherever drought was applied and seaweed biostimulant was not used, we found there is a significant decrease in the microbial population ranging across whether you say Stereobacter, then Actinomedura, Rubobacter, Alicyclobacillus, whatever. It's very important, and this was metagenomic studies. Even we tried to do some culture, petri dish culture, and we found that the use of biostimulants, they actually at 5 and 10% level, we found that it helps in improving the this CFU, this counts in the soil. And this Bacillus subtilis, Bacillus licheniformis, Pseudomonas fluorescens, these bacteria which got improved, they are actually beneficial soil bacteria. I mean, if one wants, one can always look into the paper that is published in a good journal. So there were certain unique bacteria identified that were collected from the biostimulant treated soils, which were absent where the drought was applied. So ultimately, it improves the soil microbial biodiversity, which gets degraded under drought conditions. So I will not go too much into detail. So I was also telling about the life cycle assessment (LCA) studies, and we also published this in very good journals. How we can mitigate the climate change using the seaweed-based biostimulant, and basically it is a policy decision which the governments have to take. In order to influence the government policy into using the biostimulants, wherein I mean, you can at least save a part of the chemical fertilizer by using this low carbon footprint organic products. So these are the papers one can go through, and the idea is like I think it is 118.6 kilogram carbon dioxide equivalent per kiloliter of the biostimulant. Earlier, I think I said 18.6. It is 118.6. So these are some of the papers that can always be looked into. Then we also found improvement in microalgal productivity. This is a chlorella variabilis. So this is microalgal culture. The use of biostimulant, the seaweed biostimulant, that is a macroalgal biostimulant, they can increase the dry cell weight as well as the productivity also. Now, coming to the last part of my talk, which I would try to cover in a short period of time, what is in the biostimulants because of which we get such beneficial response. Till date, there have been, uh, I mean, hearsay or general understanding that they have a lot of plant growth hormones, quaternary ammonium compounds, macro and micronutrients, amino acids. So these were the, I mean, understanding one had uh, till now. But I mean, but all of them are in concoction in different proportions. So we have actually done a elaborate analysis of all these biostimulants from different seaweeds, and we found that really this indolacetic acid, that is auxin, zeatin, and gibberellin, they are found in significant amounts when we use a simple water extract or a simple press, cold press. You get a lot of these plant growth hormones, which actually also gets degraded when you process them at a higher temperature. But then even if you process them at higher temperature, then also one can find effect. One can do alkali extraction, one can do acid extraction, or whatever the way, hot water extract. So there are different types of extraction techniques. Right now we are concentrating on which are the constituents which are lost, or which remain present. So we found that this aqua sap is the Kappa ficus sap, and Ascophyllum is the cold water species. These are the commercial ones, and we tested in terms of amino acid, and we found that even the tropical ones, there are several amino acids which are as high or even better than what is present in the cold water species. So it's like comparison of different seaweeds which we are right now also doing. One important thing is this Capoficus alvarezi, even though it thrives in the seawater, which is full of sodium chloride, 3%, it does not take that much sodium content inside it. What it takes is selectively absorbs potassium. So this potassium can actually also be a good source of biopotassium for the plants, and one can actually extract. We have developed technologies to produce potassic fertilizers from seaweeds as well. Now, apart from this, we have done high-end state-of-art liquid chromatography hyphenated with high-resolution mass spectrophotometry, hyphenated with I mean splitting, that is MS also, and we found there are several other bioactive compounds which are also reported to have bioactivity in plants. For example, kinetin, myoinositol, phosphatidyl myoinositol, sphinganin, sphingolipids, dodecamide. So these are all having some antibacterial activity, and as we also see, not only for the plants, they are also important for animals also. So that's why I mean we found improved effect on the animals also. So seaweeds they can as well be used for nutraceutical purpose also. That is something which we are also doing. So not only this, there are several other compounds also which we found using this state of art techniques. Actually, we are a chemical institute. We are chemical research, so we have access to several state of art instrumentation techniques. That's why I mean one can mingle biological and chemical research with ease over here. So that's how I mean we also compared them for different types of seaweed biostimulants. We found difference in total flavonoid content, ascorbic acid content, total phenols content. Even the speciation of the polyphenols were also done, and we found that different seaweeds like kappa 5 Ka is kappa 5 alvarezi and SW is sargassum whitey. Ka is a red seaweed and SW is a brown seaweed. And we found that it is like the content not only varied, but there were certain chemicals which were absent in case of one seaweed when compared to the other seaweed. So it's also like different types of seaweed will contain different types of constituents which also differ in their bioactivity. So that's why I mean we also tried an experiment wherein we tried to combine the red and brown seaweed, and we found very good results also. So this last part, last three slides which I am going to show is this has been recently published in one of the journals. I think it is Algal Research, wherein we really tried to zero in what are the bioactive constituents that is present in our biostimulants. We used a combination of copper, brown and red seaweed, which at applied concentration at foliar level, the concentration of which has been tried elsewhere in the literature, wherein they used exogenous isolated synthetic compound and found some good results. So this is the table about that. For example, in literature, exogenous concentration of gallic acid has been found to enhance photosynthetic rates and regulated the shikimic acid pathway. So somebody reported, and we found that our biostimulant at the applied concentration at foliar level, it also contains concentration that is comparable to that. So 7 to 11 micromolar or ppm. So the essence is that these biostimulants, the constituents in biostimulants, they may act at micromolar levels. So likewise, for example, catechin hydrate, people have applied zero point 0.1 ppm and had got good results in upregulating the perception and biosynthesis auxin hormone production. And our biostimulant contains I mean more than 10 times or 16 times of it. So that means the concentration used can be lowered, because what I'm talking about is people have found exogenous application of single compound beneficial, whereas our biostimulant contains a combination of several such compounds in physiologically active concentration. If one wants to go through this paper, I have explained in great detail in that paper. So these are the compounds which we found bioactive, which were also present in the seaweed biostimulant. So this is the paper what I was talking about, and this reads scientific basis for the use of minimally processed homogenates of red and brown seaweeds. This is a fairly recent paper published in 2023, I think 1 or 2 months back itself. So This is something that I had been doing in Germany in collaboration with Germany. This is something that was done on tomato plants, wherein we try to encapsulate this biostimulant in uh, this some polymer, and we found that the tomato roots, this is tomato roots, they tried to engulf the uh, this encapsulated biostimulants, whereas where these encapsulates did not contain the seaweed biostimulants, they even did not touch. They were as scattered as they can be in tomato, and we found improvement in, I mean, flowering also and growth parameters also. So likewise, one very important thing is roots. Invariably, we found that in any of the seaweed biostimulants, significant effect on root physiology is there. So we are still working on these aspects, and we have had, I mean, received several awards for such studies. So for today, I think I had only 1 hour, so I am exactly completing this 1 hour. So now I again hand over the session to Dr. Erika, so that if one has any questions, I can address them. Thank you very much. Thank you for patient listening. Thank you very much.
So thank you very much, Dr. Gosh, for this very interesting presentation, for giving us a great overview about seaweed extract. Now, as you anticipated, we will have a Q&A section. So I just wanted to remind that it's still possible to submit your questions using the Q&A box at the bottom. We have already received a couple of questions, so I will start immediately with the first one. The first question is, based on your experience, have you conducted trials to compare Gracilaria edulis versus Ascophyllum nodosum versus Eclonia maxima?
Yeah, good question. But we have only compared Carpophycus alvarezi with the Ascophyllum nodosum, and we found them comparable. Studies are yet to be conducted for comparing Edulis and Eclonia. So I am actually having some project that I'm going to do, wherein I will be in fact analyzing all the constituents of all the seaweed biostimulants or the seaweeds that are used for making biostimulants any part of the world. So this is something that needs to be done, and but for Carpophycus and Ascophyllum, we find comparable results. So both the formulations using Carpophycus as well as the this Ascophyllum, we found good results.
Okay, thank you. And another question. Thanks for the interesting presentation. Based on your experience, have you observed the variation on seaweed extract changing the harvesting time as reported in the scientific literature?
Yes, not only the scientific time, even the place of collection also, the same species, they will vary. Harvesting time as well as season. So there are seasonal variation as well. Harvesting time, of course, because seaweeds are polysaccharides. I mean, they yield polysaccharides, and the composition will differ at different stages. So one needs to actually do a very calculated study, a systematic study, and we found that for Carpophycus, 45 days is the best harvesting time in order to get biostimulant as well as the Carpophycus yield at economically, I mean, viable levels.
Okay, thank you. We have just received another question. So we don't have a lot of time, but if you can try to answer to this, if you could explain how you measured the carbon footprint in more detail.
Okay. Uh, we used the German model that is called Gabi, and we used the EcoInvent database. And carbon footprint was actually measured by tracking all the processes right from seaweed cultivation till biostimulant production, wherein all the processes were quantified for their carbon footprint. Individual, processes were done, and then they were modeled into this GABI software. After which we found out the not only the carbon footprint, but several impacts across several environmental categories were also done. For example, I mean, like human impact on the sea, or what is the ecotoxicity, and several other parameters were also done. And one of them was this global warming potential. So this was modeled using the GABI software, using the data that is generated at industry level. So industry level also we tried to collect all the inputs, like for example, how much is the energy that is being consumed, what is the transport, what is the fuel that is being used for transportation from production site to the retailer side. So all these things were actually done in great detail at industrial scale. If one wants, I have also given in this presentation, uh, I mean, one paper that was published in 2015 that gives the detail about the life cycle assessment, how it was done. Everything is there in that paper. You can go through it.
Okay, thank you very much. And we have finished our time for today. And thank you again for your answers. I would like also to thank you again for the nice presentation. And I would like to thank everybody for attending the webinar. And please, if you still have questions, submit them on biostimulant.com website, where there is a specific section called Ask the Experts, and our scientific committee will get back to you with the answers. I would like to remind you that the recorded version of this webinar will be available soon on biostimulant.com and on our social media. So stay tuned, have a nice day everybody, and I hope to see you on the next webinar. Thank you again, bye.
Thank you. Thank you very much. Thank you. Thank you, all of you.